Sravneet Bernini and Bradley Smith with a news report at the top of the hour on April 20th, 1988. A new study from Boston University Medical Center suggests that DNA methyltransferase, an epigenetic enzyme, has been recently implicated in certain tumors and developmental abnormalities.
Researchers have discovered that the increase of methyl tags in cancer cells is due to highly efficient enzymes found in these cells called DNA methyltransferase 1, or DNMT1 for short. Let's go now to one of the lead researchers on the project, Cody Carandish, and his associates Skylar Holtman and Chelsea Rubis. Cody, can you first explain to us what exactly epigenetics is?
Right. Brad, yes. Epigenetics is a modification process that's used by plants and animals. And using that, they can change the function of their genome without actually changing the genetic code. One of the most common ways they do that is by adding methyl tags, which are a form of reversible covalent modification. They add them to the DNA sequence of certain genes. For a gene to be translated, its methyl tags need to be removed, but whenever the cell needs it to be turned off, it adds methyl tags to the certain parts of it.
So, Cody, can you explain a little bit more about the structure of DNMT1, please?
DNMT1 is a monomeric type protein that has an active site which allows it to bind to each of the two complementary strands of the DNA. One of the domains of this molecule, which contains a zinc, binds the unmethylated target dinucleotides. This protein selectively methylates genes that need to be shut off.
So how exactly does DNMT1 do this?
Methylation of DNA by DNMT1 occurs primarily on the cytosine of CpG dinucleotides, which are repeated palindromically throughout the DNA of specific genes. DNMT1 is a methyltransferase, and it uses S-adenosylmethionine, or SAM, as a source of methyl groups. Hemimethylated DNA is the primary target of DNMT1. Hemimethylated cytosines and cytidines are flipped out of the DNA helix, and a cytosine residue on the DNMT1 nucleophilically attacks the carbon 6 of a cytosine, and that breaks up the pi bond conjugation of cytosines. This allows a concerted attack of methyl groups on the SAM molecule by a transient carbanion of the cytosine. Following this, the addition of the methylated carbon is deprotonated and it forms a new double bond as the DNMT1 leaves.
Where and when does this methylation take place?
Genomic methylation takes place during early development and undergoes program alterations during cellular differentiation. The DNMT1 enzymes are responsible for methylating DNA on cytosine at the 5-carbon position. With this, DNMT1 is located at the replication and methylated CpG dinucleotides in a new synthesized strand.
Why are the findings of this research significant to people?
Well, Brad, when methyl tags are abnormally added or not added to genes, it affects normal cellular development and can lead to the development of tumors and certain developmental abnormalities. Our team discovered that cancer cells have a higher number of methyl tags than normal cells, as well as a higher level of expression of DNMT1.
Well, that certainly sounds like an important find, but that's it for the report. Coming up next, at the top of the hour, Bigfoot. Is he real or does he owe you money?
She took back voicemail because he's got some missed calls. So.
What?
That's pretty good. Hey!
Is it like this, or like, is it weird? Should I sit this way? Or is it like the table? I don't know.
I don't think I'm going to care about that.
Should we have worn ties?
I don't have any ties.
Cytosine at the carbon. Said that word wrong. Alright, I'm so sorry. I'm just